And so we're having to subject the cells to some rather harsh conditions. This is probably the most hazardous laboratory because we're going to be cooking up on the ring stands for an extended period of time. When you guys were cooking media, it was pretty quick. When you're doing acid-fast staining on top of a beaker, the water's sitting up there boiling, you have to keep staining them for 10 minutes. So it's kind of lengthy. Be careful working in the laboratory. Okay, so the acid-fast stain is a direct stain, and it's a differential stain. Direct stain we just had a question about on the lab quiz. If it's a direct stain, it means that the stain, the colored portions of the stain are cations. They're going to stick to the cell and not to the glass. So they're going to color the cell and leave the background looking white. But what's a differential stain? The gram stain was one, and this one is also differential. It's a stain that's going to cause the cells to look different, even though you treat them all the same way. So when we did a gram stain, we made our smears, we air dried, we heat fixed, and then we stained crystal violet for 60 seconds, Ram's iodine 60 seconds. We decolorized by watching the color move and then rinsing when it stopped, and then we counterstained with saffron. So we did all these things to all the cells. If you did the gram stain properly, your gram positive cells came out purple, and your gram negative cells came out pink. How many of you actually looked at the mix? Were they pink and purple? They were supposed to be. Most people just went and looked at their morphological unknowns. It's like, come on, you got it? You want to see what these cells look like because it's part of the process and because it tells you something significant, which then I'm going to test you on. Okay, so it's going to cause cells to look different. Tonight's, sorry. So tonight's differential stain is the acid-fast. The cells are going to look different, again, because they are different. And once again, it has to do with wall composition. This kind of gets confusing because all acid-fast bacteria are gram-positive, but most gram-positive bacteria are not acid-fast. If they're acid-fast, they have high levels of a wax-like lipid called mycolic acid in their wall. Diagrammatically, it looks something like this, although this is deceptive. I think this was illustrated where there was limited space, because here you have this cytoplasmic membrane or cell membrane shown up as this great big structure, and they're showing that peptidoglycan is thin. It's not. Acid-fast cells have a gram-positive type wall. Their peptidoglycan is going to be thick. And then they have mycolic acid, this waxy-like lipid on the surface. Well, this wax is water resistant, and it tends to be also stain resistant. Consequently, these cells are very difficult to stain. They don't want to take up a stain. When you do a gram stain with cells that are acid-fast, they tend to be purple, but not as purple as you would expect them to be. They're kind of purple fading to white. So this wax-like mycolic acid makes the cells resistant to staining. That is when you use traditional methods. Consequently, we're going to use non-traditional methods. We're going to cook these guys. We're going to treat them rather severely. Once you get the stain in there, then it's hard to get it out. And so we say that they are color fast, even when you expose them to acid, and that's why we call them acid fast. Color fast means color doesn't come out. You're probably familiar with color fast fabric. You can wash it and the color stays with it. Well, these are color fast in acid, and that's what makes them acid fast. For reasons that I'm not really sure about, when we talk about bacteria being either acid fast or not, you don't ever see it written as positive, negative, like the gram stain. We say they're acid fast or they are not acid fast. The other thing, as I said, most gram-positive bacteria are not acid fast. So if you look at our examples in the room, these guys are gram-positive. These are gram-positive. These, all of those are gram-positive. Come in here. These are all gram-positive, all down this side of the room. When we get down to here, and these are acid-fast. And here's some more gram-positives. And there's another gram-positive. None of those are acid-fast except these. So lots of gram-positive organisms are not acid-fast. But all acid-fast organisms are gram-positive. They just don't stain very well. Now, because these things are very resistant to staining, we're going to cook them, we're going to subject them to heat, and we're going to use a very potent stain reagent. When we did our basic stain initially, our cheek cells, we used safranin and crystal violet and methylene blue. Crystal violet was the most potent stain. We stained for 10 seconds. Carbon fuchsin, which is the one we're using tonight, is more potent than crystal violet. So we're using this substance called carbon fuchsin. It's made with basic fuchsin, the dye, and it's dissolved in phenol, which is carbolic acid. While we're doing the heating of our acid-fast stains, we will run the fans in here to pull the phenol out because it can be quite irritating. The steps are basically like the steps in the gram stain. You're going to put on a primary stain. The primary stain is the carbon fuchsin, and it's going to color all the cells fuchsia. It's a kind of a pinkish red. It's a very potent stain. And then, and we have to do this with heat, so we're going to put them on top of a beaker with boiling water in it, so they're subjected to steam. I've been told that you can do an acid-fast stain in a microwave, but the only microwaves we have in this building are used for people's lunches, and I think they might not like it if we put our acid-fast cells in there, so we won't. There is a variation to this technique. It's called the Kenyon's acid-fast. And I don't think the color is as good, so we're not going to do that either. We're going to do a traditional acid-fast. Takes a cooking treatment, 10 minutes to let the stain really get in there. Those cells are going to be very red. Then you put a decolorizing agent on. So this doesn't require a mordant. Instead of getting a mordant to bind the color to the cells, you cook it in there. When you decolorize, you're going to be using acid alcohol. Do not use the decolorizing reagent in your gram stain set because it won't work. This is acetone alcohol. It'll pull the color out in a gram stain, but it will not remove carbon fuchsin that's cooked in. So if you decolorize with acetone alcohol, everything comes out red. You're going to decolorize with this. This is acid alcohol. I think it's ethanol with about 5% concentrated hydrochloric acid. So it does have concentrated hydrochloric acid. Hydrochloric works better than nitric. We tried it with concentrated nitric acid and it was way too quick and students over-decolorized. But the process is going to be a lot like you had to do with the gram stain where you dribble the color, you dribble the decolorizer on, you watch the color move, and be careful not to over-decolorize. So if you do this right, you're going to take the color out of the cells that are not acid-fast, and the ones that are acid-fast will stay red. So then you need a counter stain. The counter stain we're going to use is methylene blue. The methylene blue is going to color the non-acid-fast cells blue. So a gram stain, your colors should be purple and pink. In an acid-fast, the colors should be distinctly blue and red. If you see purple in an acid-fast prep, you didn't do it right. This is an illustration that's on the website. This is showing Mycobacterium, which are the acid-fast bacilli, although admittedly our Mycobacterium cultures produce a lot of cells that are very short rods. They almost look like cocks. They're supposed to be bacilli. Meanwhile, what's on here with it is Staphylococcus, and the Staphylococcus they're not acid-fast, they're going to stay blue. Remember, both of these are gram-positive, but only the Mycobacterium are acid-fast. And the acid-fast stain is typically used only in a clinical setting where somebody suspects that they might find acid-fast bacteria. For example, if you were looking at a sputum sample where you had a patient that you thought might have tuberculosis, that's usually what it's associated with. The procedure's on page 71. We're going to be working with beakers. We'll use 400 ml beakers, so fill them at least half full of water. And you're going to put them over the burner, and then we have these wire screens that we set the slides up on top of. These things are messy, so be careful with them. They're also potentially a little bit hazardous. At one point, somebody had gone around and put a layer of some kind of plastic and material on the edges so they can't stab you, but most of that's gone, so they can stab you. And they're covered with stain, so if you handle them very much, they're gonna put stain on your fingers. Anyway, so we set that thing on top of the beaker and then put our slides on top of that. So we want to get our water boiling. Ignore that test tube that's in there for the moment. To make your preparation for the acid-fast stain, you're going to just make 2 smears. So at one end of the glass, you're going to make a thin, uniform smear using mycobacterium. The mycobacterium cultures are on plates. They're in the white tray here. I'm going to add to this because this particular mycobacterium that's out here is Mycobacterium smegmatis, and the other mycobacterium we have is Mycobacterium fly. So we have 2 different species. They look pretty much the same. Both of them, because they have mycolic acid in their walls, they're kind of waxy and hard to work with. When you try to make a smear, they don't want to let go of one another. The cells tend to clump and you have to kind of mash them. Don't fling them into the air. These are potentially pathogenic bacteria because they are acid-fast. Anyway, so kind of get them to break up in that loop full of water to make as much as you can a uniform smear. Once you get them thoroughly mixed, then pick up a little bit of staphylococcus and put the staphylococcus in the smear. The staphylococcus are on slants. So you put the mycobacterium in first and then put the staph in. Try to make your smears not super thick. At the other end of your slides, you're going to use your morphological unknown. So again, that's a loop full of water. Mix some cells with the loop. You can make it into a U shape so that you can distinguish your mix from your morphological unknown. As I said, the mycobacterium cultures are on plates. The staphylococcus cultures are on slants. Those are bacillus. I don't know what those are out there for, but I'll get this staph out. Those are left over from earlier. So mycobacterium, again, the ones that are in the plates right now, they just say mycobacterium. They are Mycobacterium smegmatis. The ones that are in the incubator currently say Mycobacterium fly. And then the slants will say staph. So you allow your smears to air dry, and then heat fix them like we did before. Pass them through the flame with Bunsen burner, smear side up. Once you've done that, you're ready to begin your staining. So you're going to put the slide on top of the screen, smear side up, and you're going to cover each smear with a very small piece of paper towel. I can't emphasize enough, small. Keep your paper towel size so that it just covers the smear. Don't take a big hunk of paper towel that's then hanging off the edge of the slide. Because if you do that, when you put the carbofusion on it, it's going to wick off and spill all over the place. And we don't want that to happen. That carbofusion is very potent. So keep them small. When you put the carbofusion on, you want it to look wet. It's fairly important that you make sure your beaker is situated on the screen, or on the wire guide, so it's not tipped. You want your screen level. You want to keep your slides level. You're going to be working usually with 2 beakers per table. I guess you guys could do 3 if you want to have 2 slides per beaker. Either way. Anyway, just make sure that everything is level because you're going to put a lot of liquid on here, and you want to make sure that this stays wet during the staining process. But try not to let it drip off of there. So keep your little paper towel straps little. Using extreme caution. Carbofusion, which is about that color, is exceedingly potent, and if it gets on something, it probably won't come off. Or it won't come off unless you treat it really quickly. If you look at these counters, which are kind of green, you'll notice pink spots randomly scattered about. Those pink spots are carbofusion. Because people aren't paying attention, if you get carbofusion on the table and you leave it there for any length of time, it's there. We've tried everything to get that stuff off. The barkeeper's friend and all this stuff. It doesn't come off. Microscope number 24 has pink spots on it. That's carbofusion. You cannot get them off. So if you spill it on the bench, get your acetone alcohol and put it on that stain immediately. If you do that, then you can wipe it off. Don't leave it there, please. Yeah, watch out for those pink spots. Okay, so you're gonna let the smear stain for at least 10 minutes. While it's staining, don't let it dry out, so you have to watch it. As it begins to dry, the carbonfusion will begin to take on a kind of a gold sheen. If you start seeing that iridescent color, put some more liquid on. Don't let it dry up. Because if it does, when you pull the paper towel off, it'll take the cells off the glass, even though you heat fixed. So after you stain for 10 minutes, you rinse thoroughly with water. The paper towel scraps will go in the sink, but it's okay because there's a bunch of them in there already, and because there's a piece of mesh in the bottom to prevent paper towel scraps from going down the drain. And everybody has been doing this all day today, so there's paper towel scraps all over the place. They were supposed to put theirs in a beaker, but they didn't. Now, when we decolorize, you're going to use the same procedure that we did with the gram stain. Where you hold the slide up, you dribble the decolorizing reagent on, watch the color move, and when it stops moving, rinse. The difference is you're using acid alcohol, not acetone alcohol, not 3% KOH, which is also in the basket. Read the labels. So you're using acid alcohol for your decolorizing. As I said, the acetone alcohol won't work. So your carbonfusion is in the green tray, your acid alcohol is in the green tray. After you've finished decolorizing, rinse thoroughly with water. You want to stop that decolorizing reagent from working, so really give it a good rinse with the water. And then counter stain with methylene blue. The methylene blue has to stay on there for at least 60 seconds. After the methylene blue, again, rinse with water, then you're ready to air dry and view. When you view these, remember, focus with 10 and then go directly to 100x. Go to immersion oil, skip the 45. You can't see, you can't focus with it. Now, the other thing we're doing tonight is an endospore stain, and we're actually going to do 2 different endospore stains, one of which will require that we do the staining under the screen while the acid-fast stain is going on top. So endospores, we know they're dormant structures. They're formed by a lot of bacteria, bacillus, painobacillus, lactobacillus, etc., etc., etc. They look like this in the diagrammatic representation. Endospores. What you see here are endospores stained with what's called a malachite green stain or Schaefer-Fulton method. The cells are pink because they're colored with safranin. The spores are kind of turquoise green. They're stained with malachite green. Now, endospores are like acid-fast cells in that they are resistant to staining. It's hard to get endospores to stain. This is why when you see endospores in a gram stain, they're typically white. Sometimes they have a tiny little bit of pink color to them, but they're very light. If you had a slide and all that was on it was endospores, or let's say they were exospores because your cells have all degenerated, the spores are hard to see. They just look like kind of a pink mash of something on there, really light. So this is typical endospores. If you do a gram stain, they show up as white spots. Well, so they're going to be similarly treated to the acid fast. We want to stain them, we're going to cook them. There's 2 different methods for doing endospore stains in the lab manual. One says you're going to cook them for at least 10 minutes. The other says you can do this process more quickly, but you have to be careful. You're going to actually hold your slide over the flame of the Bunsen burner until the liquid on it comes to a boil, and then get it on. So it's a little tricky, but it does work. Anyway, we have to cook them. We're going to be using malachite green in the method where we're just boiling the material on the surface of the slide. We're also going to be doing what's called a Dorner method, and the Dorner method requires that you cook the cells in the reagent for 10 minutes. So we're going to do this Dorner method. What we do is put cells in a glass tube, a little bit of water, and then put the carbonfusion in. So we're staining them in here. This goes into the beaker underneath the wire screen where you're doing your acid fast on top. So it saves time. We get to do 2 stains at the same time. So this is showing cells stained with the malachite green or Schaefer-Fulton method. Again, the endospores are going to be green or turquoise, exospores also, and the cells are pink because they're stained with saffron. If you do a Dorner method, then you're gonna see cells that are stained not, because this is a combination of direct and indirect. So in this case, the spores are stained, and they're gonna be very bright pink, stained with carbonyl fusion. The cells are white because bacteria are transparent. And the way you can see them is your background is colored. That's an indirect stain using nitrous. So this is a combination direct, indirect preparation, the Dorner method. Lots of fun. So I'm going to put the specific recipes up on the board for these. And I'm going to, oh, I know, so the bacillus are out there for the endospore stain. Sasha said these bacillus cultures had lots of spores, but many of them were exospores. I don't know how thoroughly mature the ones that we had out on Tuesday would be, but I expect them to eat better, and I'll put those out too.
Staining.
Problem with doing endospore stains, you can't look at a culture on a slant or on a plate and know if they are or are not making spores. And so what we try to do is have kind of a combination of cells and hope that somebody's making spores. So again, these are the bacillus. The mycobacterium cultures are on the plates, and the staphylococcus cultures are also on slants. So pay attention to labels. If you pick up a tube and it feels cold, it's staph. So our bacillus are either room temp or incubator temp. Staph or cold.
Depending on how late it is. The morphological unknown is like just a data sheet. They just figure that out. Like we're supposed to be filling that out. You knew that?
Hmm?
You knew that shit?
Do we have to draw it? It's due next week. I looked at it today and I was like, I found it before class.
So
I
every
even told you guys, I think, when I came
station
in.
or every table, not
I think I
every
told you
station, every
guys.
table will have
I thought I did.
2 plates of mycobacterium. As I said, the ones that are labeled mycobacterium fly are mycobacterium fly. The ones that just say mycobacterium are mycobacterium smegmatis. Then you probably- They look about the same anyway.
They just need to take
Now, the other thing that's gonna happen, I'm gonna have to use all 3 boards. So as we're going through the procedures, you may need to move a board. Feel free to do so.
It's still recording. Huh?
Now, if I can have your attention just for a moment, relative to what I'm writing up here, small smears, because if your smears are small, it takes less stain reagent to color them, and you can keep your paper towel away from the edge of the slide if you're doing an acid fast. Uniform, because if you have chunks, those chunks are gonna decolorize at a different rate than individual cells. So you're still watching color move while your individual cells are being overly colorized. So thin, because you don't want chunks. You wanna be able to see individual cells. So small, uniform, thin smears, one piece of glass. Again, the first one, you're gonna put mycobacterium in there. Mix it around, make sure that you've got it mixed with the water, and then add stat. So this is a mix. On the other end, your morphological unknown. In the shape of a U, so that if you accidentally drop your slide in the sea, you know which smear's which. Air dry, and heat fits. If you don't do this, when you try to look at your smears with the oil immersion, the cells come off the glass and start floating around, and that's not okay. Besides, we want them dead. Okay, so again, when you get ready to do your staining, you want to have water in the beaker so that the water's boiling and you have steam coming up. Then you're going to put these little paper towel scraps on. It's easier to just kind of lay the towel scrap down and then cover it with carbon fusion. Put enough on there that it looks wet. You want it nice and wet. So you're going to stain for 10 minutes. And after that, well, then keep it wet. In this case, to keep it wet, you have to keep adding carbon fusion. You can't put water on there. Then you're going to rinse. And your paper towel scraps are going to go in the sink. And then you're going to decolorize. Remember, acid alcohol, not acetone alcohol. Acetone won't work. So again, watch the color move. When it stops moving, rinse thoroughly. Plenty of water to make sure you stop the decolorizing action. And then counterstain with methylene blue. This is 60 seconds. And then rinse. You're drying view. So you're going to again view with 10x first to focus, and then 100x. And then you're going to record data, which is color. They're either red or they're blue. You're going to record the result, which, as I said, is not positive, negative. It's acid fast or not acid fast. And your conclusion. Does that wall have mycolic acid? Yes or no? Okay, so it's kind of like a gram stain. You've got a bunch of things you have to record. This is all part of your morphological unknown assignment. So people keep asking me about those illustrations. Do we turn these in? No, until you get to the assignment, and then yes, because you have to turn in pictures. So if you're sketching them as we go along, it's a lot easier when you have to actually do your illustrations. So far, for your morphological unknown, you have an indirect, which tells you your size, shape, and arrangement. You have a gram stain that tells you they're either gram positive or gram negative. They're either pink or they're purple. Tonight, you're going to find out, are they acid fast? If they're acid fast, they're red. If they're blue, they're not acid fast. And then you're going to find out if they have endospores. I'm going to put the Dorner method endospore stain here so that you can see both of these because you want to do them at the same time. If you want to begin, you have your mycobacterium and your staff. You can go ahead and get your slides and get started on the acid fast. And I'll just put the Dorner method prep up here. And then we need our meters. We don't have a lot of meters.
I knew I should have been sketching it. I just fucking have it. I knew I should have been sketching it. I just fucking have it.
So please, no more than 3 beakers per table.
Okay, you two can do one and then we'll do one. You down?
So?
We'll do one and they'll do one.
Yeah.
Should I keep recording? Something writing.
I'm gonna get you a gloss slide.
Yes, please. Thank you.
I need 2 slides. Actually, 3 slides.